Now that we've introduced the sarcomere, let's see how its structure allows skeletal muscles to contract. In order for a skeletal muscle to contract, the muscle's sarcomeres must contract. When a sarcomere contracts, the distance between the Z-lines becomes shorter. The lengths of the individual filaments stay the same, but they move past each other and the region of overlap increases. The I-bands, regions of only thin filaments, shorten. And the H-zone, the region of only thick filaments, disappears. The sliding of the filaments occurs because of interactions between the thin filament actin strands and the thick filament myosin heads. Let's look at what many scientists currently believe are the molecular events that occur during skeletal muscle contraction. The myosin head takes two shapes, a high-energy conformation and a low-energy conformation. An ATP molecule binds to the low-energy conformation. When the ATP is hydrolyzed to form ADP and a phosphate, the head changes shape into the high-energy conformation. The high-energy myosin head binds to a specific site on an actin molecule. ADP and the phosphate are released, and the myosin head changes shape back to the low-energy conformation. The change in shape pulls the thin filament toward the center of the sarcomere. Each thick filament has around 350 myosin heads, and each one can go through this cycle about 5 times per second. Therefore, the filaments can move past each other quite rapidly. A skeletal muscle cell stores enough ATP for only a few contractions. Most of the energy for contraction is stored in phosphagens, substances that can supply a phosphate group to convert ADP to ATP. In vertebrates, the major phosphagen used is creatine phosphate. This is why some athletes take creatine phosphate supplements to enhance their muscle performance. Since muscle cells store energy for muscle contraction, our body needs a mechanism to keep the muscles from contracting all the time. Muscle contraction is inhibited by the tropomyosin of the thin filament. Tropomyosin blocks the site on actin to which the myosin head binds. The location of the tropomyosin is controlled by a set of regulatory proteins called the troponin complex. For the muscle to contract, the tropomyosin must be removed from the myosin binding sites. When calcium ions bind to the troponin complex, the interaction between the troponin complex and tropomyosin is altered. This causes the tropomyosin to change its shape, exposing the myosin binding sites on actin. The calcium ion concentration in a muscle cell is controlled by a specialized type of endoplasmic reticulum called the sarcoplasmic reticulum. A signal from a motor neuron telling the muscle to contract causes the sarcoplasmic reticulum to release calcium ions. Tropomyosin inhibition is removed and the muscle can contract.